We often get hurt in our daily lives, and our vessels are getting damaged every single time, just like this. The holes in our vessels get closed immediately by platelets and blood coagulation factors. This activity starts by a protein like this. This is referred to as primary hemostasis. On the other hand, the proper size of blood clot is governed by the complex cascade of secondary hemostasis. It's important to note that hemostasis shows nonlinear dynamic, where a small wound under threshold will not generate blood clot. Here we designed a system called BIOMOD that makes artificial clotting to cover wound. BIOMOD combined two breakthrough technologies to develop B's aggregation system. One is the chemical reaction system called PEN-DNA toolbox, and the second is the DNA-anchored microbeads called swarm robots. When the concentration of the released DNA is above a certain threshold, the microbeads will start to aggregate, forming artificial blood clot. BIOMOD is inspired by the associated DNA released by damaged cells around the wound. BIOMOD consists of two parts. First part is the thresholding with PEN-DNA toolbox, and second is the clot formation using swarm robots. In our system, we prepare template DNA for amplification and three types of enzymes. When the input DNA from dead cells presents, it will hybridize to the complementary template DNA. After hybridization, the signal DNA is elongated by polymerase and followed by nickase cut on a specific recognition site of the elongated strand. This is repeated over time, amplifying the concentration of signal DNA. How to suppress the signal of DNA from small wound? Strand alpha in this system gets degraded by exonuclease and inactivated by drain template. The decreasing rate of strand alpha is nonlinear because of combination with exonuclease and drain. The concentration of strand alpha in the equilibrium state of amplification and degradation is the threshold. After the synthesis of strand alpha is converted into a linker DNA by the same chemical reaction scheme of strand alpha amplification. I will show you the mechanism of B's aggregation. The linker strands produced by PEN-DNA toolbox is the complement of the strands anchored onto the B's surface, intermediating the aggregation of B's. In this way, we came up with BIOMOD, which conduct clotting in nonlinear dynamics like secondary hemostasis.